In this video, we will have a look at bacteriophages and how we can use them in molecular biology. Bacteriophages, or short phages, are viruses that specifically infect bacteria. They are widespread and exist wherever bacteria are found. Phages come in many shapes and forms. One of the most well-known are the T4 phages, which resemble a lunar lander. Structurally, they have an icosahedral head or capsid, which contains the genetic material. The head is connected to a tail that allows them to interact with their bacterial host. Phages are the most abundant entity on Earth. For example, a single milliliter of seawater contains between 10 and 100 million phages, which kill about 20% of the microorganisms in the ocean every day. That is why they rank among the deadliest biological entities in the world. However, the interplay between bacteria and bacteriophages also regulates these ecosystems. As a result, bacterial populations remain balanced. Let's have a look at how phages infect their host. Phages do not move with purpose. Rather, they rely on random interactions to find their bacterial host. Once a phage comes into contact with a bacterium, it attaches to the cell wall using specialized tail proteins. This binding occurs through electrostatic interactions and is influenced by factors like pH and the presence of certain ions. Phages are remarkably specific in how they recognize their host. They bind to unique structures on the bacterial surface, such as lipopolysaccharides, teichoic acids, proteins, or even flagella. Phages degrade the bacterial cell wall and inject their genetic material, DNA or RNA, directly into the cytoplasm. In some cases, this genetic material is circular and can mimic a bacterial plasmid. Once inside the host, the genetic material of the phage can follow two pathways depending on environmental conditions: the lytic cycle or the lysogenic cycle. Let's have a look at the lytic cycle first. In the lytic cycle, the phage hijacks the bacterial machinery for protein production, forcing it to produce new phage particles. Initially, the phage recognizes a bacterium and injects its genetic material into the cell. The genetic material replicates inside the host and becomes the blueprint for assembling new phages. If a critical mass of new phages accumulates within the host cell, the bacterium will simply burst open. The newly produced phage particles are released into the environment and can infect new bacteria. Phages that exclusively use the lytic cycle are called virulent phages. In contrast to the lytic cycle, phages that undergo the lysogenic cycle integrate their genetic material into the bacterial genome. After injection of the genetic material, the phage DNA integrates into the host DNA and becomes a so-called prophage. In this dormant state, the phage does not destroy the host. Instead, its genetic material is replicated passively along with the bacterial genome as the host divides. Under certain conditions, for example stress or changes in the environment, the prophage can be reactivated. When this happens, the phage genes are expressed, triggering the lytic cycle. Again, new phage particles are produced and released after the cell bursts open. Due to these capabilities, bacteriophages can also be used as a tool to transfer genes from one bacterium to another. This process is called transduction. Transduction is based on the fact that during the lytic cycle, the bacterial DNA is broken down to smaller fragments by phage enzymes. These fragments can be randomly packed into the phage particles. These transducing phages, which carry bacterial donor DNA, are released together with normal phages during bacterial lysis. When this lysate is used to infect a new bacterial strain, a small fraction of the recipient bacteria take up this host DNA. However, if the donor genes do not recombine homologously with the chromosome and become permanently incorporated, they are lost. Since recombination is a relatively rare event, selection is required. The property of phages is used in genetic engineering for the transfer of DNA segments. By including a selection marker, such as an antibiotic resistance gene, only bacteria that successfully integrate the desired DNA through homologous recombination will survive during selection. An example of a phage that is frequently used in such experiments is the Enterobacteria phage P1. The genome of phage P1 consists of linear double-stranded DNA of approximately 93 kilobases. It encodes for around 100 proteins. Phage P1 specifically infects Escherichia coli cells and is a so-called temperate phage. That means it can undergo both the lytic and the lysogenic cycle. In the lysogenic cycle, P1 does not integrate its genome into the host chromosome like many other temperate phages. Instead, its genome exists as a low copy plasmid within the bacterial cell, which makes it interesting for genetic engineering. The picture shows the structural components of P1, including its icosahedral head, which contains the DNA. Its long tail plays an important role in recognizing and attaching to bacterial hosts. After infection and incorporation of the host DNA, about 0.1% of all phage particles will carry fragments of the host DNA. The P1 phage can also carry 2 or more genetic markers within the same transducing particle. This so-called co-transduction occurs when the markers are located close enough on the bacterial chromosome. Since the size of the packaged host DNA is limited to approximately 100 kilobase pairs, only nearby genes can be transferred together. P1 transduction was originally developed to determine the localization and arrangement of genes on the E. coli chromosome. The proximity of the selection marker to the gene of interest also determines the efficiency of the co-transduction. Genes that are closer together have a higher likelihood of being co-transduced into the recipient strain. This forms the basis of the following experiment.